Thank you. So we're gonna shift gears a little bit. Pictures are a freezing moment for me. Yes. So we're gonna shift the gears, and instead of talking about patients and specific clinical groups, we're gonna talk about basically all of us, so healthy participants, and how the gut health can affect decision making. And we are constantly making decisions, and decisions impact our well-being, our financial future, our social relations. Actually, they impact our society as a whole. So it's a very important topic. And human decision making is very complex. I focus on a very specific type of decision making, which is risk-taking behavior. And risk is can be defined as a situation where you have uncertainty, but you kind of have an idea of the probabilities. So the probabilities can be measured. A good example of that would be, for example, if you're gambling or playing with a dice. But there are also examples in your daily life. So how do you choose if you're going to bring an umbrella? You can check the forecast and see what are the probabilities of rain in that day, and then you can decide if an umbrella is a good option for you, or if you're Dutch, raincoat, or maybe if you're Dutch, actually, you're always going to carry an umbrella and a raincoat. So that's how you should follow. But if you don't live in a place where it rains all the time, that should be a good example of risk-taking behavior for you. And we know a lot about decision neuroscience already. We have been studying this for decades, and we have some ideas of specific brain areas that are involved in decision making. And some common denominators are the intermediate prefrontal cortex, dorsolateral prefrontal cortex, anterior cingulate cortex, insula. And those brain areas, they have been reported in several studies using neuroimaging, for example, and also other techniques such as non-invasive brain stimulation that can affect the regions, those specific regions, and see how it affects human decision making. But I'm proposing a new player here. So what about your gut? Is that a possibility? Maybe your gut is also affecting how you decide. And we heard a lot about the gut-brain axis today. I think that's going to be something we're going to hear a lot about also tomorrow. So I'm going to try to not overlap too much. But we know that a balanced diet can improve your gut health. And we know that the gut can affect your brain activity, it can affect your mood, it can affect many other aspects of your cognition. And or you can also use probiotics or prebiotics or postbiotics. There are many options, and we are hearing a lot about all of them, to try and rebalance this communication between your gut and the brain. So there are some effects of probiotics or effects of changing your gut microbiota that are more well established in the literature. For example, there are studies that report that there is an effect on sensitivity to pain or effect on mood and stress reactivity. And there is a growing body of studies that show that there's also an effect in cognition and brain activity. And talking specifically about risk-taking and decision-making, we have some studies that used animal models to demonstrate that by affecting the gut microbiota, you can affect risk-taking behavior in animal models. So what you see, for example, is that germ-free mice, they express more risk-taking in specific tasks, and this can be normalized, neutralized by normal colonization. You also see a reduced risk-taking after you give probiotics to this specific strain of rats that are considered a model of depression. So those are rats that would initially take more risks, and you can reduce risk-taking by giving them probiotics. But what about humans? So when we started this line of research, there was nothing yet on decision-making. But we had a lot of evidence already coming from neuroimaging studies, and we saw that resting state studies indicates differential activation in areas that are important for decision making. So we see a lot of overlap there, which shows us that not only the animal models indicate that affecting the gut microbiota can lead to changes in decision making, but also what we saw in humans until that point indicated the same thing. So we see a lot of repeated effects on the salience network and the default mode network, and those networks they overlap heavily with important brain areas for decision making. So we had some questions to start with this. So does the gut-brain axis affect the human decision making, and how is this effect happening? We combined probiotics. We opted for probiotics. Maybe we should try other, perhaps fiber and prebiotics next time. We combined that with a test that we developed to evaluate risk-taking behavior, and we looked at risk-taking behavior in a neuroeconomics perspective, so looking at economic decision-making. So it sounds like a very big gap because we're talking about economic financial choices. But we wanted to start exploring from there, and that's the first step we took. We also looked at time preferences. So time preferences, basically what it means is, are you willing to wait a little bit more to get more money tomorrow? Or are you willing to wait a little bit more to get a little bit more money in a month, for example? This is very important for our financial well-being. So besides risk-taking behavior, we checked time preferences, and we did a placebo-controlled, double-blinded study. And what we saw is that actually participants that received the probiotics manipulation for a month, 4 weeks, perhaps that's also a factor, they had higher patience and they were willing to wait more to get a little bit more money. Actually, the way that we did, we discounted the current payoff, or they could wait and get the entire money later on. Usually we are very impatient. That's something that's natural for human beings, that we're very impatient and we prefer the immediate reward instead of waiting a little bit more. But we saw, and this happens because we have what's called discount rate. So we are willing to lose a little bit of money, like pay a little bit of money to get the reward immediately. And we saw that the group that received probiotics actually were had lower discount rate. They were willing to wait a little bit more to get more money. Looking at risk-taking behavior, it was a little bit more complicated because those placebo effects. So what we saw is that risk-taking behavior was stable in the group that received probiotics. What does that mean? When you receive some money, we humans have a very weird effect that in economics is called the house money effect, which means that if you have money in the bank, usually you are more willing to take risks. That's a distortion because the money that you have in the bank doesn't affect at all the odds of you getting money in a gamble. So you should not do that. That's not really a rational decision making, but it happens. And we saw this house money effect in the placebo group. Based on the money that they got in the intertemporal choice test. So the more money they had, the more they took risks in the placebo group. However, when we looked at the probiotics group, this group was stable. So that indicates that somehow the probiotics were creating a protective effect, which can be associated to many different cognitive processes, but we believe that we're talking about a better emotional regulation. But we wanted to understand this a little bit better. We went forward with our study. And first of all, these findings are actually in line with what we saw in the animal models. So when you have a distorted baseline, so we're talking about these depression models where the rats already take more risk than they should, then you have a reduction in risk-taking behavior. That's exactly what we saw in humans. So when you are taking more risk than you should, you have a reduction, relative reduction in risk-taking behavior. Okay, so we still have a lot of questions, right? Those effects are a little bit entangled and we don't know how this is happening. So the next step was to check only risk-taking behavior, isolating that. And what we saw is that when you isolate risk-taking behavior, so when you have a stable baseline, when the placebo group behaves as you expect, you have actually an increase in risk-taking behavior. However, this increase in risk-taking behavior was paired with higher response times and no impact at all in choice optimality. What does that mean? It means that the higher risk-taking behavior was not associated with higher impulsivity, which is a very important factor in when you're talking about decision making. So it's a more deliberate choice. Higher response times are associated with more deliberate choice, and there was no impact in choice optimality. So if you are taking risks without calculating the odds of your returns, you might decrease the quality of your choices. And what happens usually is that since we are naturally risk averse, we avoid even bets and gambles that could be beneficial to us because we want to avoid risk. So there are circumstances where risk is a positive outcome. Risk is desirable. It will be the rational choice that we would naturally avoid, and the group that received probiotics were more willing to take risks. This again goes in line with the theory that this group could handle the emotional signals that gambling would give to them. So emotional regulation seems to be a key point here. So from these steps, what we saw is that when you have a distorted perception of risk, when you have a distorted baseline of risk-taking behavior, you would have the probiotics giving a stabilization effect, so a protective effect. When you have a stable baseline, you would have an increase in risk-taking, but an increase in risk-taking with deliberate and high-quality choices. The how is a very complicated question here, because as we saw, there are many mechanisms that are involved in this communication between the gut microbiota and your brain. And we are still studying this. We are implementing a lot of different studies in a big research line funded by the Dutch government. And we are adding several neural techniques so we could gain some insights on how this communication is happening, what's happening in the brain that's leading to these behavioral effects, and perhaps how this communication literally is happening from the gut to the brain. There are many pathways that are involved in this communication, in the gut-brain axis communication, and one big candidate for this communication is the vagus nerve. So the the composition of fibers of the vagus nerve is actually mainly efferent, so it mainly goes to your brain. And we see studies, for example, the effects that we saw when you colonize the rat that was germ-free, and then the rat starts behaving normally in terms of risk-taking behavior, you can reverse those effects with a section of the vagus nerve. So when you interrupt the vagus nerve, you would revert to the effects before the colonization. So this will be a potential way of finding out what's the main pathway that communicates these changes in the gut to the brain. However, in humans, I don't think any ethical committee would allow us to section the vagus nerve. We could try, but I don't think that's a good idea. So we went in the second best way, which will be to stimulate the vagus nerve. And the vagus nerve is connected to many different cognitive functions. And again, we go back to this common denominator of emotional regulation. There is a lot of overlap between the areas that are activated when you stimulate the vagus nerve and areas that are involved in emotional cognition. So what we did to test if the vagus nerve would be a potential main pathway for this gut-brain axis communication, we did a study with non-invasive vagus nerve stimulation. And we saw that actually the effects obtained with the probiotics and the non-invasive vagus nerve stimulation, which was in this case applied in the ear, they're actually very, very comparable. With a small difference, and that's a very interesting difference, is that when you stimulate the vagus nerve, we actually got an increase in choice optimality. So that's the only difference. Other than that, the effects are very comparable. So that means that you can emulate the results, apparently, at least the behavioral results that you have when you give the probiotics protocol compared to the vagus nerve stimulation. We also wanted to investigate the mechanism, so we started looking into the ventromedial prefrontal cortex. The ventromedial prefrontal cortex is an area that's involved in many things. If you are from decision neuroscience, you probably heard that this area is involved in valuation. If you are from neuroscience, you probably heard about Damasio and the somatic markers hypothesis. It's an area that is still a little bit mysterious because it's a deep brain area that's hard to stimulate. So we have a lot of insight from neuroimaging studies, very few insights from non-invasive brain stimulation studies. And those non-invasive brain stimulation studies are what allow us to make causal inferences. And this area will be a good candidate for mediating this effect from the gut to the brain because it's an area known for integrating the somatic signals. So it's an area known for integrating these bodily signals into higher level cognition, such as decision making. So we started small. We started by trying to deactivate this area in your brain, very small, very simple. So we used a new development in transcranial magnetic stimulation. We used a new coil that allowed us to stimulate this deeper brain area. And we actually managed to do that. What we saw is actually that this area and the dorsolateral prefrontal cortex are very interconnected. But the point here was to test the technique so we could modulate risk-taking behavior by doing that. And we saw an increase in risk-taking behavior when we deactivated the ventromedial prefrontal cortex. So we went to the next step, very optimistic, thinking that everything will be very simple. But again, we could increase risk-taking by deactivating the ventromedial prefrontal cortex, but we could not find a significant interaction with the probiotics. So although promising, the ventromedial prefrontal cortex still not the answer here. Our next step here will be to run a study with the probiotics in the MRI so we can have an idea of a more targeted area to use with TMS, so we can try again to deactivate this specific area that we are hoping to identify, and then be able to modulate directly in the brain this effect from the probiotics. So the idea is that we turn on the effect by giving the probiotics, and we can turn off by deactivating the specific brain area that will be pivotal for this effect in the brain. We also looked at EEG results. So we started by replicating. There are only 2 studies out there so far using EEG on the gut-brain axis. So it's a good topic for everyone that wants to investigate. Using EEG is a very promising technique as well because it's affordable. The results are very straightforward. So we saw significant results in alpha power, beta power, and theta power. And those, of course, alpha power, we are relating that to attention, and we are talking about reductions in medial prefrontal cortex and increases in left, the LPFC. So we're talking about also executive control here. Beta power, we are investigating this specific frequency band because it's related again to emotional regulation. So that's one of our hypotheses. And theta power is related to risk-taking behavior. So we saw effects in medial prefrontal cortex, but you can see that this setting was very restricted. But looking at those specific frontal areas, what we saw is that after receiving probiotics, participants had a reduction in medial prefrontal cortex activity, in alpha power and theta power, and also in beta power. And we saw increases in left LPFC, in alpha power, and increases in beta power as well. So the follow-up will be to investigate the same effects with a larger setting and perhaps looking at different brain areas. And this is what we have so far. So this study is happening. Actually, we have one of our interns here present. This study is happening now. So we have participants going through this 4 weeks protocol of either probiotics or placebo. And what we see now is actually this resonance of medial prefrontal cortex. It's something that's happening again. But we also see some temporal effects compared probiotics to placebo, and some occipital effects. I think that we are pretty much in line with what we saw already this morning. But these are only preliminary effects, and this is only 10 participants, and we already in each group, and we already have some quite striking results. But this is the next chapter, and many of the questions that we have are still to be answered. So we have still a line of research to go through, and perhaps in the next edition I can present to you the final results. So the final conclusions of those studies so far are that the healthy gut can influence economic decision-making. And this is a big claim, and it's something that we actually started in a very exploratory way. But if you think about these findings that we got in terms of economic decision-making, it's a very it has a very striking impacts for society. If you think that people that are in lower economic that have lower economic means, they actually have access to low quality food, highly processed food. What we call the Western diet is more used by low income populations. That would lead to poor economic decisions. So thinking about a wider range of impact of these studies, we can perhaps, if we can establish these effects with certainty and also with larger samples, we can think about societal suggestions of or changes in policies that could perhaps focus more directly on gut health. Specific results that we had were this effect on intertemporal choices, more patients and willing to invest in future options, and riskier yet more deliberate decisions without impacts in choice optimality. We have this potentially protective effect against choice distortions, which again, we believe that's related to emotional regulation, and electrophysiological effects on alpha, beta, and theta power. But the mechanisms are still unclear. Although we have these similar results with vagus nerve stimulation, we don't have an idea if these similar effects would also be reflected in brain activity. So far, we only know about behavioral effects. And we still didn't find our ideal TMS target, so we can try to infer some causality there. And that's it. This is just the beginning. If you have any questions, please shoot away.
Thank you very much. Any questions from the audience?
Maybe about the mechanism that you said that they are still unclear, but I'm also wondering if you're trying to measure some parameters of the stomach, like motility index or some. Because, yeah, you know that probiotics should influence stomach activity, but in which way? And second question is, should also be, what are the information that from the stomach goes to the brain? What is the brain sensible to?
Yeah.
So, I don't know if in general you have, some hypotheses or some planning study in the future on this.
Yeah, not specifically with gut motility. That's a very interesting question, actually. That's something that we could think about integrating into future studies. So far, the plans that we have are to look specifically into the effects in the brain, because there is a lot of literature already on resting state, but we don't know exactly what happens during decision making. This new study that we're running with EEG is also on, we are looking at resting state, but we're also looking during decision making, because the effects could be specific for specific scenarios. So it can be that the effects that we see in terms of behavior only because during specific challenges, during specific emotional challenges, the brain would react differently. Other hypothesis that we are currently studying is that the probiotics manipulation itself would affect food preferences, and then you would have a more global effect. We ask our participants always to keep stable diets, and we have questionnaires to check that. Usually they report stable diets, but again, we're talking about questionnaires, so we are not really sure. So one hypothesis that we have is that this mechanism that we see in economic decision making is actually a carryover effect from the gut microbiota trying to reach a stable diet, so risking. How can I define that? So the level of risk that you would take, or if you want to consume more now or later, would be actually an economic spillover effect of how you would choose food-wise. So we are looking at food preferences to see if people are, if this manipulation is leading to choices of healthier food items that will be less risky food items, and also in terms of variability, so that will be also less risky, or items that have lower caloric weight. So it can be also that you're willing to invest for your future also food-wise. So our hypothesis is more in this direction, that the effects that we see in economic decision-making are a spillover effect of a natural mechanism that we would have to keep homeostasis and for this gut bacteria to keep a stable, ideal environment. But that's so far just a hypothesis. Yeah.
What is not clear to me? Is there also an investigation in parallel of anxiety and stress related to these choices?
Yeah, so first of all, we try to control for stress and mood. So we have some questionnaires and also arousal. We also try to measure that. So we have questionnaires for those. We never had any significant effects in those questionnaires after the probiotics manipulation. This is the third time that we are running studies with this same manipulation. We use the ecological barrier by Winklow. Perhaps that's. So in this sense, I believe that's not the case. Other thing that we have taken care of is that the task that we use has a wide variety of levels of risk. So you would expect that you have a wide variety of excitement as well. So you have trials that are very, very easy and trials that are very, very risky. And the effect is actually very global. It's very widespread. So my sense from the data that we have is that that's not the case. That's not about arousal. Full disclosure, the first study that we did, we tried to manipulate stress with the cold pressor task as well. The cold pressor task, we had zero effects of the probiotics on the at least on the declaration of stress after the cold pressor task. So we decided not to follow this line. So perhaps the only stress that they have is the gambling itself.
And a question more about the sort of the, at the beginning of this research line. So it's that fMRI study where you show that after probiotics, there is a differential activation in some subcortical networks. And so from there, the idea that, okay, because there is a different activation, some of those networks may influence risk-taking. Mm. But in that study, what was actually we go back to the placebo, right? Because I'm thinking if it's like an empty pill as compared to one that it's probiotic rich, one sort of does activate some motility, more or less goes into that direction, right? Some gastrointestinal activity that then is reflected via the gut-brain axis to the brain. The other one is empty, so you don't see anything. But does it necessarily mean that you have an influence on those cortical subcortical networks that may influence choices? So what is the hypothesis?
That's a very good question. So more recent studies, actually, there's a very large study by Esther Arts, that's one of our collaborators in Dantas, that what they did, they found correlations between specific bacterial strains and the activity of specific brain areas in this resting state. So it's possible that there is an effect of gut motility. I think that we're talking about a very complex mix of effects. We're talking about immunological responses. We're talking about differential production of neurotransmitters. We're talking about a very complex network here. And I don't discard at all that gut motility is a big factor here. But there is some evidence of correlation between activity of specific brain areas and specific bacterial strains. In the case of our studies, we are not looking at specific strains, so we use always this probiotics composition that's a mix of culture. But that will be an interesting question as well. If you can have the same effects with single strain probiotics, or perhaps with fiber, we can so many ideas from today. Yeah.
And the effect is immediate, so they take the pill and then.
No, we don't know actually.
Because
Yeah, we
because also with EEG, right? I mean, you're looking at some signatures that are super high resolution, but also like as soon as you put the cap and you start recording, you're expecting already some effects, for example, changes in cortical excitability or something similar, or what is also the timing of the effects? You wait or something?
That's a good question. So what we do, and perhaps I should have added this here, what we do is that we have the same procedure in the first and the second session, except for the intake of probiotics or placebo. So it's a double-blinded. We actually don't know which one they're taking, but they start taking the probiotics or placebo after the test. So we have the first measurement without any difference between the groups, and then we have the second measurement after 4 weeks. It is possible that there are effects already during these 4 weeks. We don't know. Participants don't detect any difference. We didn't have any reports. So I'm sure that I'm taking probiotics. I'm sure that I'm taking placebo. But this is also because we are using a placebo that's produced by the same lab. So it's identical in color, in taste, in presentation. So perhaps this helps a little bit. It's not always possible. Yes.
Actually, now I have two questions because this one raised another one. Do you think that maybe the placebo effect might be regulated a bit if the tested person are only people that work in the field, let's say? Because sometimes I think that common people say we take whatever, decide to participate, I'm taking a pill, and therefore there might be an effect, even though they are just probiotics, so physical effect won't be detected, of course. But maybe that's something. So maybe restrict the cohort of patient only to people that actually know that this pill won't make anything, even if have probiotics inside. Physically, I mean,
Okay, no.
physical effect.
So you're talking about the first study that we had, the house money effect in the placebo group,
Yes.
right? So that effect was, our statistical analysis showed that the effect is directly connected to how much money they made. So if they didn't have any extra money, they didn't have the house money effect. It was literally a distortion for how much money they had. And then when we replicated without this extra money on the side, then we didn't have this effect anymore. It's interesting also to think that people that would have more experience with gambling or more experience with financial decision making could react differently. We hear that they want to be very rational. In practice, the decisions are the same. Because we have this test that we use is 250 trials. They have 125 unique trials in random order. And actually, people think that they are going to be very rational, but they end up acting very quickly and being very intuitive. So it doesn't change much how much experience they have, but that's a good question. Yes.
And
Then we,
Yeah.
yeah.
Well, first of all, maybe I should revise my statement that probiotics are not needed by a healthy population, especially if they influence your mental functioning, let's say.
Right.
Because I'm really intrigued by the fact that you see an effect on decision making, and you also referred already on the effect on making food choices, maybe. Because we are always struggling with a specific population with a low socioeconomic position, let's say, they are not making the best health choices. So do you think this might be worthwhile to see if probiotics can affect their choice making for the healthy foods?
Absolutely. That's the point now. So our idea with this research line is literally to establish this strong connection so we can suggest public policies that would implement perhaps in schools or perhaps in work, they say that in English, mensa, canteens, exactly, in work canteens, to implement perhaps a richer diet. Of course, the best idea is to have the Mediterranean diet, which has equivalent effect than the probiotics. If it's not possible, probiotics or prebiotics or postbiotics, now we have to think about it. But to implement gut healthy programs that can potentially lead to better, not only dietary choices, but as we saw, better economic choices. Because it's also a problem that we see that populations that have lower income, they are more subjected, for example, to gambling addictions. So this is a vicious circle that we want to understand, and perhaps in the future break, we gotta hope. Yes.
Yes, final
So,
one.
do I understand correctly that you didn't find a relation between mood and the change in risk behavior or in
Exactly.
right?
Yeah.
I'm asking because we recently found the opposite. So we did find that risk aversion actually modulates how people mood-wise improve from probiotics.
Oh.
So there seems to be kind of a, I don't know, a weird interaction.
Yeah, it can be. But one thing is that we're looking at mood following a questionnaire. So it can be that that's how we're measuring it. Other thing that is interesting in our studies is that we use the global preference survey to also evaluate risk preferences. So if they're risk takers or they're risk averse. And people declare differently than they behave. So that can also be something that can be that's the effect that you see in declarative risk taking is not exactly how they behave. Declarative risk preferences is not their risk taking behavior.
Yeah, and perhaps also depending on their mood. Right?
Absolutely, yeah. We take a lot of care also to keep the conditions very stable in terms of avoiding any excitement in the lab or trying to or anything that could change the participants' mood, because decision making is so subtle that any small difference indeed can change how they decide. So this is one thing that we try to control the best that we can. Big fan of your work, by the way.
All right. Thank you very much again.
Thank you.